In this screencast, we are going to take a look at the process of DNA replication. Before we talk about DNA replication and that process, it's important to review the importance of the structure of the DNA molecule, of the DNA polymer. And Watson and Crick were the first to finally put together a model and describe it in their 1953 article, the simple one-page article in the journal Nature. They described the double helix architecture of the DNA molecule. And in reviewing that, remember, we know a few things about the components of each of the nucleotides that are involved, and we know that there are 4 different types of nucleotides in DNA. We talk about them with just their letters a lot of times as A and T and C and G. But Watson and Crick actually noticed, and they're quoted as saying, that their description of the double helix molecule actually also suggests a possible copying mechanism, or in other words, a way that DNA makes more of itself, or in other words, how it replicates itself. In DNA, we want to know that DNA has a direction. And remember that a nucleotide is the subunit of the polymer DNA, made up of a phosphate group, a nitrogenous base, one of 4 kinds, and then a 5-carbon sugar called deoxyribose. And it's important to be able to count the sugars and know that that's the way that they are always, the orientation is always the same. So from 1, 2, 3, 4, and 5. And we're going to talk about how these link together, and it's important to know these 2 distinct carbons, the 3' and the 5' carbons. Recall that the DNA polymer has a backbone that is alternating sugars and phosphates. And we also remember that each strand of a DNA double helix has a 5' end and a 3' end. And this refers to those carbons we just identified, the 3' end and the 5' end, the 3' end and the 5' end. And the 2 sides, the 2 complementary sides of the DNA polymer are called anti-parallel, which means that the 5' to 3' strands run in opposite directions of one another. It's important to note that the 2 main types of bonds in the DNA polymer are hydrogen bonds and covalent bonds. Now, the covalent bonds, which is good enough for us, the covalent bonds are very strong bonds. In other words, the sugar and the phosphate are covalently bonded to one another with a very strong bond. The hydrogen bonds, if you remember, are rather weak, and this is going to be an important note when we actually talk about how DNA makes more of itself or how it replicates. Using Chargaff's rules, Watson and Crick also identified the importance of distinguishing between the purines and pyrimidines, and that the base pairing rules are always going to be a purine and a pyrimidine. And so we've got the purines always bonding with a specific pyrimidine, and the distinction, A always binding with T, and C always binding with G. It's often asked, the question comes up by students like, well, what if C would actually bond with T? And as you can see that molecularly, it just can't happen. There are 2 bonds available between adenine and thymine. There's 3 bonds available between guanine and cytosine. That's asking the same thing as, well, what if my house key did actually start my truck? Well, they aren't chemically matched. They aren't the puzzle pieces that are going to fit together. It only works in these ways. In copying DNA, recall that we called it a semi-conservative process. And in that semi-conservative process, that just means that each side is going to be a template for a new strand of DNA. So really, the daughter strands, the new strands of DNA, or the new molecules, are going to be hybrids. One strand, or 1/2 of the double helix, is going to be the old original strand, and the other half is going to be the new copied strand. And there's really two main steps in DNA replication. We're going to keep it that simple. The double helix is first unwound, which makes then two template strands, which are noted here with the blue strands. And those are available for new base pairing. Then new nucleotides form base pairs with the template strands, and those are linked together by their specific bonds. The DNA template strand is read in the 3' to 5' direction. So DNA replication is basically just two steps: unzipping or unwinding and unzipping, and then adding new nucleotides. But there's more than a dozen enzymes and proteins that participate in DNA replication. We're going to narrow that down to the main players. And in DNA replication, we have what are called replication forks. That's the location where the DNA strand unzips and where replication takes place. And you can notice that the replication directions that we'll talk about a little more specifically can occur in two directions. It's always going to occur in 3' to 5' direction. So again, the first step is the unwinding of the DNA. The unwinding of the DNA, the main player here in an enzyme you need to know that does that is the helicase, is the name. It unwinds this DNA helix. So it's shown here in the yellow. Now that helicase, then, as it unzips the two sides, and those sides are then stabilized by what are called single-stranded binding proteins. And they're basically what hold open the replication fork. And in the second step of replication, we're actually going to build the daughter DNA strand. We're going to use the original as a template, and using nucleotides, we're going to, complementary nucleotides of course, we're going to build the complementary strand of DNA. The second big player in this DNA replication that you need to know is called DNA polymerase. There's actually a couple different types of DNA polymerase, but we don't really need to distinguish between them. DNA polymerase is the enzyme that binds the new nucleotides into place. So this is just showing you again, remember that we are reading the original template strand from the 3' end to the 5' end. The DNA polymerase is what is going to be adding the new nucleotides. The new nucleotides and this process is actually energized by a couple extra phosphates that the bonds are broken off of the nucleotides as they bind to the existing nucleotides. But again, it's built from the 3' end to the 5' end. So while we talk about this, the direction of the DNA strands, remember that the original DNA strand, a double helix, is anti-parallel. That means it has two strands that run in opposite directions of one another. Well, that comes into play and is of importance during DNA replication. So the new strands can only be laid down in a certain direction. DNA polymerase can only build onto the 3' end of the existing DNA strand. So it's always going to build from 3' to the 5' end on the template DNA. So that's okay in what's called the leading strand. In the leading strand, we will just add on to the 3' area and build in a continuous direction, okay, with the growing replication fork. Well, since the replication fork actually started at this end, what's called the lagging strand can't be built in the same direction. Instead, there are small fragments in which, at the replication fork, the new daughter strands are laid down in a complementary fashion in the opposite direction. So they are laid down here. Then we'll jump to the next section and lay them down here, and then to the next section and lay them back. So kind of it jumps forward and then goes back, but according to the 5' to 3' for the new side, or the 3' to the 5' for the original template strand. These fragments are called Okazaki fragments, and then they are put together by a third enzyme called ligase. So this can be really confusing to get the whole 3' and 5' ideas straight. Just remember, on the original template strand, it is always read in the 3' to 5' direction. What that means is, because of their strands being anti-parallel, is that the new DNA strand, the complementary strand, is always laid down from a 5' to 3' direction. You want to keep reviewing that so that you can keep this straight. So another enzyme involved is one that is called primase. Primase is the enzyme that kicks this whole process off, and it starts by building onto the 3' end of the original template strand. And it's actually just a single base of RNA. RNA is another nucleic acid that we will discuss in much more detail very soon. But it serves as the starter sequence. It's the starter sequence for DNA polymerase to know where to lock on to start adding the new bases. So part of its job, along with adding new bases, DNA polymerase, kind of a special kind, but DNA polymerase is going to replace that RNA primer with the proper nucleotide of DNA. At the other end of the process, we have telomeres. And what those are just repeating non-coding sequences at the end of the chromosomes. They're kind of like a protective cap. And they know where that's like the end of the DNA polymer or the end of the chromosome. And the enzyme that's involved here is called telomerase, and it extends those telomeres, and it adds bases at the end of the molecule. This is a great diagram to stop and to pause and to review the enzymes that we've talked about, the strands that we've talked about, the difference between replication on the lagging strand versus the leading strand, to cement your understanding of the directions of the 3' to 5' direction on the original strand versus adding new bases in a direction of 5' to 3' with the newly built daughter strand of DNA. Pause the video, check it out, study it, review it, learn it, know it. This whole process is just amazing. There is editing and proofreading that is going on. DNA polymerase also does this proofreading, corrects any little mistakes that might be made, and they put the proper bases. They replace them with the proper bases. If there seems to be a mismatched base, DNA polymerase makes the correction. This happens throughout life, the life of an organism, and although errors can occur, it's amazing that the error rate is as low as it is. Not only is it accurate, but it's very fast. In the bacterium E. coli, less than an hour to copy 5 million base pairs, and it just has a single chromosome, has DNA that is found in a ring. And for that to divide into 2 identical daughter cells, remember from our original cell cycle unit that we have to have the synthesis of the DNA, of the genetic material, before the cell can actually split. Now, human cells, they copy its 6 billion bases and divide into daughter cells in only a few hours. So it's remarkably fast, and it's remarkably accurate. So I hope you take the time to review the process, review the main players that are involved, which enzymes are involved, and the direction of the copying of the DNA molecule in this synthesis of a DNA molecule. It is extremely important that we know the process. It seems like it's kind of on a context right now, but we will, as we talk about the rest of the central dogma of life, we need to understand the structure of DNA and how it replicates itself.